Hello, this is our Olympus VS110 high throughput slide scanner. Think Google Earth for microscope slides. It allows you to zoom in and out and navigate around a virtual digital version of your slide. You can then send that data to someone anywhere in the world for them to view, to analyse or diagnose from. Instead, if you happen to physically send them a delicate and sometimes irreplaceable glass slide. The microscope works in the same way as the dot slide microscopes in our outer room, which David will tell you about, but it has extra bells and whistles. Firstly, it can automatically scan up to 100 slides in one go. And secondly, it can work with fluorescently labelled slides. In here is the microscope itself. The slides sit in a pair of cassettes that live in here. And in here is the robot that moves the slides from the cassettes onto the stage to scan them and back again. Here comes the finger, it picks up the slide and it carries it to an alignment jig at the back of the microscope to make sure it's perfectly square onto the finger. It then takes a macro photograph of the entire slide. Picks it back up in a perfectly aligned orientation and moves it onto the stage of the microscope, where it then gets scanned. You can hear the stage moving as it takes the individual tiles of the image.
Here are some examples of the types of slide scans we can get from the system. This is part of the human gut, stained with the chromatic dyes hematoxylin and eosin and scanned at ×40. There are 1,699 individual images in the dataset. This is part of a human placenta, stained with a polychromatic stain and scanned with a ×40 objective. There are 1,938 individual images in this dataset. This is part of a human brain, which has been stained using an antibody, recognising a particular protein in the brain, and the antibody has been visualised using an enzymatic reaction which produces a brown colour. It has been scanned with a ×20 objective, and there are 2,133 individual images in the dataset. It's only when we zoom right in that we can see the antibody staining of the individual cells. And this is a fluorescently labelled sample. These are sections of a mouse eye, which again have been stained with an antibody, but this time fluorescently labelled, and it highlights a single layer within the retina. The nuclei of the retinal cells have also been stained here with a DNA binding stain, which stains in blue, and our antibody is staining in green. There are 909 individual images in this dataset. And finally, another fluorescent example. This is part of a human lung, this time stained with 3 different fluorescent probes, shown in green, blue, and magenta. It's been scanned with a ×10 objective, and there are 1,068 individual images in this dataset.